Just how when we had talked about with protein hormones or peptide hormones, glycoprotein hormones, whatever you want to call them, the receptor is kind of what determines what's going to happen downstream, right? Acetylcholine can be excitatory or inhibitory depending on the receptor type. And so when we're talking about steroid hormones, I think it's helpful to look at the generalized structure of a steroid hormone receptor because the structure is what lends it to its function, right? There's 4 domains of a generalized steroid hormone receptor, and this consists of the N-terminus, which is the variable domain. So this picture that you're seeing down here, it's running from N to C. The N-terminus is the variable domain, which means it probably doesn't do anything. Next to that, though, and this is shown here in blue, is the DNA binding domain. This consists of the zinc finger, and a zinc finger is a very important site of phosphorylation, which is a very important site of regulation. Next to that, over here in purple, you have the hinge region, which is involved in transport. Very poorly illustrated in this picture, I know, but this is where you'll find the nuclear localization signal, and in some types of other examples we're going to talk about where HSPs are kind of involved, and we'll explain that later, but that's where they'll be bound to. The next we have the C-terminus here, which is all this stuff here, which consists of both the ligand binding domain, and after we have ligand binding, or in this case hormone binding, we will have dimerization of our receptors. Okay, so there's a term that is used in your textbook that I don't quite understand their obsession with, but they used it a lot, so let's talk about it. Hormone response elements. So once the hormone receptor complex moves into the nucleus, it's going to bind to a specific regulatory sequence in the DNA. That specific regulatory sequence is the hormone response element. Usually the promoter, I would assume that they could probably also bind to some distal control elements or other deals with that. Gene expression in eukaryotes is a little bit funky, kind of weird how much bending and stuff going on there. But anyways, what's important that you understand really for this class is that it activates or it will repress transcription. So there's actually 2 subtypes of steroid hormones that are mentioned, and these vary depending upon the mechanism of how they work, right? So type 1 is going to be bind to either a soluble or receptor in the cytoplasm or a soluble receptor inside the nucleus, right? Whereas type 2 steroid hormone receptors, they just bind directly to the DNA or directly to the proteins that are involved with gene expression. So let's see if we can't have, yeah, okay, cool, pictures here. Okay, so this is type 1. Here we see the hormone here diffusing through the plasma membrane because it's lipid soluble, and then it's going to bind to a heat shock protein complex. And then upon binding there, the heat shock protein will dissociate, the receptors will dimerize, and then upon dimerization, it goes into the nucleus and you have gene expression happening. This is just 2 pictures showing basically the same thing. The HSPs here is the same thing as calling them chaperones. It should be chaperonin. I don't like that picture too much. But anyways, some examples of this would include sex hormone receptors, testosterone, estrogen, cortisol receptors, and then mineral corticoid receptors. And one of the things that I'm kind of curious about, and maybe someone could explain to me, is why are we specifically using heat shock proteins involved in this? I know that those guys are just abundantly expressed, but it's always found that quite interesting. Okay, so for type 2, this, as you can see here, the hormone goes just, in this case, they just kind of left out the overall cell here. I guess if I could include it there, that'd be the cell. This is the nucleus. The hormone goes through the nuclear pore just as is. It doesn't have any, receptor bound to it. And once then, in there, it will bind to its receptor site, and then again, activating transcription, having messenger RNA being produced. But keep in mind that they can also act to inhibit or decrease levels of transcription. And some examples for type 2 would be A, vitamin D, retinoid, and then thyroid hormones. So let's just kind of go with a quick review of the generic steroid receptor structure. Obviously there's the N-termini, and the N-termini is the variable domain, which is not very well conserved, which probably doesn't serve any function. Next to the N-termini, we have the DNA binding domain, which is conserved, which does have a biological function. I'm just going to abbreviate that as BD for binding domain. Structurally, this is made up of 2 globular motifs. That's a G, by the way, globular motif. What's important about this is that this contains a zinc finger. It's for obvious reasons made up of both zinc and then cysteine as well. Is this important? Because this is a site of phosphorylation. My handwriting normally isn't this shitty. It's just these tablets. God, I hate them so much. Okay, so next we have the hinge region, which contains the nuclear localization signal. All right, hinge region contains the NLS or nuclear localization signal, which is involved with heat shock proteins sometimes, and then the transport is the function of this guy. All right, so last but certainly not least, we have the C-terminus. And this is where we're going to have hormone binding and then receptor dimerization. Cool. Hormone binding, then dimerization. Okay, so very quickly, let's go over type 1 versus type 2. So type 1, and these guys are all differentiated based off of their mechanism of action. So for type 1, the mechanism of action for them is they bind to a soluble cytoplasmic or nuclear protein and then get to their site. And then for type 2, these guys bind directly to the DNA proteins or to the transcription initiation complex or to the co-receptor, whatever, right? So yeah, so type 1 binds to cytoplasmic proteins and then we'll have transport. So for type 2, these guys bind directly to the DNA or to the DNA proteins. It's a hormone response element. So yeah, to either co-repressors, or I guess I could have also said activators or to the transcription initiation complex.